Environmental DNA has been around now for almost 20 years as a tool. It was first detected in things like permafrost, and it has gained most of its application in aquatic systems. And so what we're looking here is to explore different ways that we can collect that DNA. The reason this is important is that it's really, really time intensive to do biosurveys. Trying to actually see an animal, to count it, or to use camera trapping relies on it being there, present in front of you. Environmental DNA is different because it lasts in the environment longer. The animal doesn't have to be present for you to detect it. So it's a really unique tool to have in the biosurvey toolbox if you can detect the presence of animals from days previously, to know they were in the vicinity or that they do use that habitat. That's a huge advantage in understanding the use of landscapes by animals. We used a pump system that pulls air through a very, very fine filter. And that filter is so small, it can actually trap DNA particles on it. What's really unique here is that two groups have independently verified that this is viable as a source. These really very early demonstrations that we can do this outside where the air is moving and the weather is present and the sun is out and the wind is blowing, and we can still do this highly successfully, is really very, very promising for this to be a technique. Within our own research project, we know we were collecting DNA hundreds of meters away from the source. And that was what was really unique about the zoo as a location to test this, because we knew where each animal was, and then where we collected the DNA, we can actually look at how far it moved. And we didn't see really any good relationship between distance from the animal and the amount of DNA we were collecting. It was certainly concentrated around the enclosures where they live, but we were able to get it hundreds of meters away and even from closed buildings. So there were a couple of buildings that were actually sealed during the period for the health of the animals. We couldn't get inside them, but the DNA was actually escaping. We were collecting it out in the environment.